Okay. Is that changing?
Now, yeah.
All right. Okay. Sorry about that. Okay. Well, here's what you missed. The global health challenges, Quantum Mission 3, and the summary of our flagships. So yeah, moving on to this is the research that we wanted to present today. So the first topic is about labeling with nanodiamonds. So this is rather than using the MV sensors as sensors, using them as labels to improve the sensitivity of diagnostic tests. And actually, we presented some work on this in the last ADAMAS talk that I did in, I think it was in 2021. And then so here I'm presenting some translational work we've done on that, applications to some different disease areas and some clinical translation. And then some work sort of in relation to that, which is about modeling the value of this extra readout sensitivity. And then Felix is going to talk about our move trying to push this towards a point of care compatible system that will then hopefully be taken forward in some clinical settings. And then Felix is going to talk about the Widefield coherent sensing. So the first thing is just to briefly introduce the challenge is in these diagnostic tests, point of care tests such as lateral flow tests, the things that limit sensitivity are things like your signal per particle. Usually you use a nanoparticle to label a biomarker of interest. So you attach your nanoparticle to a biomarker of interest, and that way you convert something which is very difficult to detect, like a protein or a fragment of DNA, into something which is very easy to detect because it is stuck to a nanoparticle with a very clear signal. The other considerations are binding rates, non-specific binding nanoparticle concentrations, and things like volume and flow rates in flow tests. One of the major issues on lateral flow tests is you have this big background autofluorescence, comparable, as you can see here, to sort of a 50 nanomolar concentration of quantum dots. And so so it becomes a signal to noise ratio challenge. So trying to get the best signal to noise ratio, which corresponds directly to your limit of detection, which then corresponds to your clinical sensitivity. So people have tried a range of different ways to get around that. Time gating, so you can separate out in the time domain. You can separate out in the wavelength domain. So what nanodiamonds, what NV sensors and nanodiamond offer is both a large Stokes shift and the ability to externally modulate. So what this means is that we take one of these tests. This is usually a sandwich assay where you have two binders binding to the target, creating this sort of sandwich structure, and we replace whatever gold nanoparticle is used in that particular whatever nanoparticle is used in that particular diagnostic. In the case of lateral flow tests, it's mostly gold nanoparticles. We replace that with a nanodiamond, but otherwise leave the test completely unchanged. And then we do essentially 1 point CW ODMR. So we're continuous exciting with green light and then applying an amplitude modulated microwave field. So we oscillate between a mixed state and a mostly MS equals 0 state. And so we have about 5 to 10% contrast, and we can modulate between those two and make the test line flash. So this is a line of immobilized nanodiamonds top to bottom, and you can see them sort of flashing at 4 Hertz, and then the background is pretty steady. And that's what it looks like in the time and frequency domains. And then you can see this sort of 1/√T scaling for the negative control. So yeah, we apply a lock-in algorithm to separate out the flashing signal from any background signal in the frequency domain. So we end up with a signal which is very specific to the nanodiamonds. This gives us roughly a 10 to the 5-fold improvement over gold nanoparticles in a lateral flow test. This is what we showed in our paper in 2020. And then in a real assay, this corresponds to a 7,500-fold improvement. We showed a single copy detection of HIV RNA, and we did a small, with a 10-minute isothermal amplification step, so keeping test times very short by having a sensitive readout. And then we showed a sort of proof of concept clinical seroconversion panel where we compared to PCR. So moving on to some more recent work translating this. And we've looked at a few different disease targets and disease areas. The first is wastewater epidemiology, and this was work led by Da Huang. So The problem here is that people would like to monitor a wastewater as an early marker of.
Excuse me, we have half of slide for a long time. With schematic of the OMR detection or DMR detection.
The slides are not moving on.
No, can you go, say, just between two slides now to click back and forth?
Okay. Do you know what?
Should you try it from your computer?
It was your 2020 paper slide there, schematic, that it ended.
It's not sure. Why is it not there? Sorry about this.
Oh, it happens. It's
okay.
I'm not sure.
If we just sharing it now.
Okay, I think it's probably best if we maybe set up Ben's computer to do this as well.
Can you see that?
yeah.
Okay.
And then we were on slide 9. We were on slide 9 when it was frozen. Sort of.
Can you see this? Is this now wastewater epidemiology?
Wastewater epidemiology, yes.
Okay, let me know if it stops again, but hopefully.
Okay.
Hopefully we'll do better. So yeah, the problem statement here is that wastewater surveillance can be helpful for early detection of infectious disease outbreaks, such as in a pandemic scenario. You can detect diseases in the wastewater rather than relying on people to be testing at home, and then that can inform sort of local policy interventions. So the problem is that this is usually done by PCR, which takes 24 to 72 hours, which can be important in a pandemic scenario. And the sort of the rapid tests for doing this are not sensitive enough because in wastewater the concentrations are extremely low. This work was led by Dar Hang, as you can see here, and Dar developed a nanodiamond assay which combined this kind of isothermal amplification with a nanodiamond readout. And ran a pilot study with samples from the Welsh National Wastewater Programme. So it uses a small sample volume of 100 to 200 milliliters. Hopefully the slides changed. Did it change? Okay, great. And what Dar showed was a limit of detection of about 7 gene copies per reaction. And you can see in the bottom right here, a comparison between the signal to blank ratio on the y-axis with nanodiamond assay compared to PCR for PCR positive and PCR negative tests. And what this translated to was 100% sensitivity and 100% specificity with gold nanoparticles. And then Dar also ran a comparison with graphitic carbon nanoparticles where he showed 80% sensitivity and 100% specificity. So this was, an effective rapid test for wastewater surveillance. The next application we looked at, this was work led by Elisa Thomas de Cruz during her PhD. This was looking at early detection of SARS-CoV-2. So the motivation here, so you can see in this plot, we have these horizontal lines indicate the limit of detection for RT-qPCR, and then the limit of detection for the antigen lateral flow tests, which were very widely used in the UK, certainly during the COVID pandemic. And you can see that because of this difference in detection limit, you lose all of this time in terms of diagnostic. You lose about 2.5 days between when you could detect the disease with PCR and the infection with PCR and when you can detect the infection with these rapid tests. So we were trying to make a rapid test that would get closer to the RT-qPCR detection limit. Again, it's the same procedure, but just swapping out the biomolecules for antibodies targeting the SARS-CoV-2 antigen, nucleocapsid antigen. And Elisa did a blinded clinical study of 53 positive and 50 negative samples in collaboration with Elena Stuli at UCLH, which is the university hospital. And what Elisa showed was 87% sensitivity and 100% specificity compared to RT-qPCR, as you can see here. And we have the limits of detection for the nanodiamond assay and for the gold nanoparticle assay marked on, and all of the samples in between are where we gain an advantage. We combined this work. We combined this analytical.
Again, sorry, let's us make sure we move to your data. We on slide.
Yeah, now there should be a table and a graph.
No table, so
Okay,
please activate.
You send me the slides. Okay, we'll switch to my computer. Sorry. This is the diagnostic window, this sort of lost time that we're hoping. So we're hoping to bridge the gap between these antigen tests and PCR. So yeah, we produced a similar test, but we switched out the molecules on the surface of the nanodiamonds and on the strip for antibodies targeting the nucleocapsid, the SARS-CoV-2 nucleocapsid protein, which is well conserved. What Elisa did is a study of a blinded clinical study of 53 positive and 50 negative samples from the university hospital, showing 87% sensitivity and 100% specificity. So you can see we've plotted on the detection limits of the gold nanoparticle and nanodiamond assays, and you can see that in this middle region here is where we gain this diagnostic advantage. Then what we did is combine that with data from human challenge trials and surveillance data to try and model the real world impact of being able to detect these lower concentrations, these lower viral loads. And what we found, so you can see here, this is your viral load infection dynamics over time, days after inoculation. This is from human challenge trial data. And then what you can see is shaded in blue is the blue dashed line is the detection limit in terms of days with the nanodiamond assay, and the green dashed line is the same for a standard gold nanoparticle assay. And the shaded areas between the 50th percentile patient, the mean patient, and the 95th percentile patient, so the patients with the lowest viral loads. And so what you can see is that we get a 2 days earlier diagnosis with nanodiamonds compared to gold nanoparticles for the 50th percentile patient, and for the patient with the 95th percentile lowest viral load, we can actually detect 2.8 days earlier. Now, also combining this with patients reporting symptoms, we show that we can detect 2.2-fold more patients detected on day 1 of symptom onset, which at the peak of the Omicron, as an example, at the peak of the Omicron wave in the UK, I think would have meant about 70,000 more people detected on the first day of symptom onset. And yeah, we published that earlier this year. So in terms of the outlook in this area, we have sort of 2 sensitivity limits. There's the fundamental sensitivity limit, which is limited by material properties, and then we have our current assay sensitivity, which is limited by the biochemistry. So in terms of the fundamental sensitivity, our contrast is proportional. So our detection limit is proportional to 1 over our contrast. So any improvements in contrast give you a corresponding improvement in your detection limit, and therefore clinical sensitivity. The same for NV sensitivity. That's the LOD is proportional to 1 over the root of the NV density in nanodiamonds. So if we can have more counts, we can get better sensitivity. Also, the homogeneity of the NV centers is important, and how much microwave and optical power we can supply. And this is a trade-off between having the best sensitivity and also having a test which is more amenable to lower resource settings, uses less power, uses less expensive components. And then finally, better spin control. So because in this test, all of our NV centers are randomly oriented with respect to our B1 field, and we're not applying a bias field, that means that we're not actually switching between a 0 and 1 states. We're switching between a predominantly 0 and a more mixed state, because we have these different Rabi rates, and we're sort of mixing all of our spins. Now, in terms of the current assay sensitivity, before we get to this fundamental limit, we're limited by two things: non-specific binding and antibody association rate. So this is essentially how imperfect the binding is, the binding that you want is, and also non-specific binding is the binding that you get in the absence of a target. So that obviously brings our noise floor up, and then the antibody association rate brings our signal down. And so that's what leads to the difference between our fundamental sensitivity of 10 to the 5-fold better than gold to our real assay sensitivity improvements, which are between 1,000 and 7,500-fold improvements. And so if we can improve on that, and that's something that we're working on, then we can get closer to this fundamental limit. The key thing there is differentiating between specific and non-specific binding. So as sort of this concern has sort of is the motivation for the work that I'm just going to briefly talk about next, which is some work by one of our PhD students, Sam Hopgood, and this is looking at modeling the value of readout sensitivity. So we can show these very large improvements in these very large possible improvements in sensitivity, but what we want to know is what kind of chemistry is required in order to fully realize that potential. So what are the properties of our binding molecules that we need in order to justify using an ultra-sensitive particle system like this rather than a less sensitive one? For example, maybe we could use the nanodiamond simply as fluorescent labels without doing any modulation, or under what circumstances does the modulation give us an extra benefit in a real assay? And so what Sam is doing towards this is looking at the various, is modeling the binding of all the various species in this test. So looking at transport and reaction kinetics, well mixing, washing and receptor distribution. So this involves just defining a set of differential equations, partial differential equations, which describe the time and spatial information of the test, and keeping track of various things like the density of available binding sites on the nanoparticles, the density of available binding sites on the test, and the available target. And so you keep so the model keeps track of mobile species and bound species, and that allows you to plot what all these different species look like in both space and time. So the blue to green is the that's the wrong way around. Green to blue is the progression forward in time in these plots on the left. And then the x-axis is the spatial dimension along the test, and then the y-axis are the various species. And so you can see that we can get a pretty accurate model for the shape and amount of nanoparticles that are bound, and so we can look at the signal, and that's what this plot on the right is showing, the signal over time. So Sam has refined the model in various ways and ended up with very good agreement between the experimental and simulated signals. That's what you can see in this bottom plot here. And so then what this allows us to do, and this is still a work in progress, but this allows us to look at the effect of things like the particle concentration and this Ka, which is the association rate of the particle, which is a biophysical constant. And look at what signal we get. So yellow is higher signals and blue is lower signals. And then we can plot this against particle concentration, and we can say, okay, in what regime do we need to be in order to get a benefit from using a more sensitive nanoparticle system? And the answer is we need to be in this regime where our particle concentration is high enough that we're in this saturation regime up here. And you can see that where the saturation region starts depends on this association rate, on this property of your binding molecule. So you need to be in this saturation region for your positive samples and for your highest positive samples, and also below your noise floor for your negative samples. So I guess it's not surprising you need high signals for your positive samples and low signals for your negative samples, and then you do get this benefit from being in this, having these ultra-sensitive particle reader. So yeah, just going to pass over to Felix, who's going to carry on with the final two topics.
Great.
Hello, everyone. Yeah, so you've heard a lot of the good work about kind of taking this from the fundamental demonstration that was initially done in 2020 and applying it to various types of assay targets and showing some improvement. But the kind of vision for this is that would provide sort of near PCR levels of sensitivity while maintaining this lateral flow test point of care format. And all of the work that you've seen previously discussed was done on this microscope, essentially. So this is a centralized laboratory system, and it's sort of like one of the pieces that we need to improve on if we really want to take this to a point of care compatible system and leverage this ultra sensitivity in maybe resource limited settings, is that we need to create a measurement system which can be deployed in those settings. So what would we need to do that? We need, all of these considerations. I'm not going to read them all out, but you need to optically initialize your NV centers. You need to be able to detect the fluorescence. You need to be able to do processing, things like that. And so the idea in the group for some time now has been many of these things you can achieve with a mobile phone. So it's kind of a portable computer. It has a camera and things like that, data storage. So why don't we try and kind of recreate this microscope and optical excitation system, but in a much cheaper format, leveraging a mobile phone. And so what some of the first work that we did on this was to basically put the mobile phone onto the microscope system. So the excitation optics are the same, the strip is the same, all of the other aspects are the same other than the detector, even the kind of integration time. And so you can see that we've got significantly more noise on the mobile phone. So it's not the ideal starting point for an ultra-sensitive diagnostic system, but we kind of persisted and moved forward with that just to see how far we could get. The other issue, as well as just the noise that's on the detector, is the kind of the bit depth of the sensor. So you have much less dynamic range when you're encoding in 10-bit versus 16-bit on the camera. And essentially, this is the system that we came up with. So we have a nice high-powered laser in here, some microwave components for generating 2.87 gigahertz, modulating that. Everything is powered by a little power bank that you would charge your mobile phone with. So the laser and the microwaves, everything is powered via that. And this is some of the time trace data for some of this direct bind assay that Ben was talking about, what you use to kind of establish your fundamental limits of detection. And so this kind of oscillation here is not noise. This is a 4 hertz modulating signal. These three are a single concentration. Then we have green, half the concentration. And then this kind of blue and gray one, the gray being your blank, kind of shows potentially why it's advantageous to have a lock-in. So if you just look in your fluorescence count rate, there's no statistical difference between these two sets of three. Whereas when you look in the modulation time trace, you can see that these three blue curves are statistically distinguishable from your blank data. So that's some of the kind of, the raw data. And then how does this compare in terms of a fundamental limit of detection? So we can see Ben's original data on the left, and then some remeasuring a new set of strips is in green on the right. So 2.2 attomolar this time is the LOD. And then on the mobile phone with the same strips is 36 attomolar. So it's a factor of around 15 worse than the microscope, whereas gold nanoparticles on the same microscope was a factor of 100,000 worse. So I think there are, we're really pushing towards, well, I think we've achieved significant improvements over gold in terms of the fundamental limits of detection. And typically you can't really reach your, as Ben was describing, you can't actually reach a full sensitivity floor anyway. So we're quite hopeful that this would, kind of lays the groundwork to performing some form of translation and pilot studies and things. So I did some measurements on the SARS-CoV-2 data sets. And this is some of the original data on the right from the paper, and found a 140 femtomolar LOD. I think this is limited by strip placement precision as opposed to actual, like fundamental limits of the system. And to kind of prove that, we worked with a different assay, one of these ongoing wastewater projects, and compared the LOD between the microscope and the reader. and found very comparable results. I mean, the caveat to this is that due to the fact that it's a very nice amplification-based assay and we're getting high target concentrations, you can also do quite well in just fluorescence. So this kind of speaks to the work that Ben was mentioning before around like, when is this ultra sensitivity necessary? Do you always need modulation? But essentially, I think this kind of proves that, you know, you can create these kind of low-cost, portable, low size, weight, and power devices and still perform NV center modulation. So we're relatively hopeful that this can move forward and we can apply it to more targets. We're currently in the process of trying to identify where the biggest value add would be for something like this in a healthcare system, which isn't necessarily always simple to figure out. But yeah, that work is ongoing within the hub. And so then, yeah, to kind of move on to quite a different project and the title of the talk, I'm aware that this has been kind of sort of pushed back to the end. But yeah, we want to essentially do wide field coherent sensing with ensembles of nanodiamonds at low or zero magnetic field. And so why would we want to do this? Nanodiamonds, as many of you in the audience probably know, are extremely versatile sensors. And there's many different like kind of hosts for NV sensors. So you can have bulk, you can have NV-AFM, single nanodiamonds, ensembles of nanodiamonds, and coherent protocols, ones where you actually create some superposition. For example, if you want to do DC sensing, it's much more sensitive to do a Ramsey measurement than it is to do a CW-ODMR. And so a lot of the most sensitive protocols involve creating some coherent superposition. And this has typically been limited to systems where you can align a magnetic field to your NV axis, split your ±1 state, and then do all your nice control with high fidelity. But that limits the types of systems that have typically been used before. So people use diamond chips because, you know, you have 4 orientations, but you can't put that inside of a cell. And also you need to, every time, say you functionalize some biomolecule on there, then if you want to use it again, you have to reset your surface every time. And then, so there's a lot of really nice work in that direction, and people are doing, yeah, great work. Similarly, people are doing loads of interesting things with single nanodiamonds in cells. However, the signal that you get from them is quite limited because, you know, it's a single nanodiamond, either an ensemble of NVs or sometimes a single NV in there. The counts can be quite low. Also, you want to align a field, you then have to track your nanodiamond and vary your field and stay aligned and all these types of things, or shift your frequency. And an alternative method is people are doing work where you have lots of nanodiamonds in some system, you measure them all at the same time and do T1 relaxometry, which can be insightful in terms of pH. And that there's a lot of interesting things you can do with that, but it's limited to measurement of noise at single frequency. So it can be quite non-specific. A lot of different things can influence a T1 measurement that aren't necessarily spin relaxation. So, and I will say we're not the only people working on this based on this kind of set of information. Faz and Shi, which was formerly Yang Feng Du's group, are doing a lot of interesting work on this as well. Well, we're doing slightly different, but we want to utilize many nanodiamonds, image them all, kind of, wide field excitation, so 500 by 500 microns, and still do these types of coherent measurements. And then we're collecting onto a single pixel, so we're getting all of those counts in one go, and then you can understand what's going on in a sample. So we're not at the stage where we're introducing this into biology. We're kind of doing fairly proof of principle work at the minute, but we are interested in applying this to biology. Eventually, you can imagine something where you have maybe 1 million cells in a field of view. There's a nanodiamond in each of them. You do some maybe radical oxygen species detection, and you get some statistical understanding over 1 million cells rather than just looking at one cell. And so that's kind of the vision, and we think there's a lot of interesting work. We're still a way off that, but we hope that this kind of, is in the right direction. So the main reason why people, I think anyway, people haven't done this previously is because you can't align a magnetic field. And so you're going from the 0 to the ±1 state. So everything's at 0 field. The other aspect of this is that because all of your NVs have random orientations, all your nanodiamonds have random orientations, your B1 is kind of spread across them. So your control rate is proportional to the perpendicular component with your NV axis. But so we've done some simulation work and shown essentially if you just have a lot of different NV centers, completely randomly distribute them. This is the kind of distribution of your NV axis relative to the Z axis. But then if you were to apply a control field along that Z axis, the distribution of your control rates actually looks quite generous because there's another sign in there essentially. And so your control fidelity is not so bad for a system like that. We want to do multi-frequency control. So this is a cross-section of our coplanar waveguide that we're using. So if you then import that field distribution as well, you kind of get something that's a bit more smeared out. But again, it's not as bad as you might think in terms of your control rates. And so we can still get quite nice Rabi pulses at 0 field with massive amounts of field inhomogeneity. I mean, they're not incredible, right? It's not going all the way back up to the top, but we can still do pi pulses and pi/2 pulses and control sequences. So that was kind of the first thing that we realized. And then we figured, can we actually do some coherent sensing with this? So the trace in black is a Hahn echo sequence, which is phase cycled. So you have one of your final pi/2s is a pi/2, the other one is a -pi/2, and then you reference that signal. So we can do a nice Hahn echo, and then we apply some arbitrary frequency here. We're showing 1 megahertz and 2 megahertz, and you can see that with this coherent superposition in this kind of system, you're sensing some AC field. It has the right frequency dependence, it has the right power dependence that you would expect for a system like this. So that was kind of a nice first step. And then we wanted to establish some sensitivities. So, if you're in this dip, you're obviously much more sensitive, but we've done some analysis kind of showing how, if you take the final average data, how does that track back, and how does it do it if you actually take each individual piece of data there. And because we're getting quite a lot of light, we have very good SNRs for very low measurement times. So you can see, we're getting quite good 10⁻² SNR for quite, less than a second. And then if you start to calibrate this to magnetic field strengths and look at your AC sensitivity, I mean, it's not the most wonderful sensitivity in the world, right? 140 nanotesla per root hertz compared to some of the ensemble magnetometers that are out there with NV centers, just because, the control isn't as good, and we're having to limit the amount of light that we're collecting to not break our detector. And also the amount of light going in is spread over a large amount of space. So our initialization times are quite slow. So it's not the best fundamental sensitivity, but we think the kind of system itself has other advantages. We're calculating these sensitivities, and then the next step that we performed was to do double electron spin resonance. So for people who aren't familiar with that, is you use your NV center as your magnetic field probe, and then you flip spins of a variety of frequencies, or you apply a broad range of frequencies. And then when you hit some target frequency, when you hit some spin that has the same target frequency, that changes the local magnetic field. And it's a nice spectroscopic technique. And we think this is interesting, in the field of Ross and various other things, to be able to, characterize spins that are in your nanodiamond. We are limited to systems which have a 0 field splitting. So this is P1 centers in the diamond. So they have a hyperfine splitting of, you can see these two peaks here, like 149 megahertz and 130 megahertz. We think that this is a very nice demonstration of the technique that we can do sensing of electron spins in our environment. Now, We can see also a big kind of G=2 line down there, and we think there's some, we can do some interesting ratiometric analysis on different nanodiamond sizes. And also we can control our target spins. We're seeing these kind of coherent P1 center DIR mutations, and at different RF powers, it's kind of the scaling is as we would expect. So obviously we would like to understand our sensitivities, and so we've done some analysis on that. And yeah, essentially we can get quite high SNR for quite low measurement acquisition times, and that's kind of the strength of this technique. There's a lot of things we can do to improve on this, so increase the laser power, basically improving our optical initialization time, which is far below saturation here. Nice, yeah, improve T2 nanodiamonds, and we want to be able to perform more sensitive protocols, so XYA, things like that, where you would increase the interaction time of your NV center with your target field. We're currently limited to just standard Hahn echo and standard DIR Hahn echo due to kind of control fidelity issues, but I think adiabatic pulses is a solution for that. However, we're currently power limited, but we're working on solutions for that. And so we predict with various with a variety of these improvements, we can get down to the low nanotesla regime, and it's probably a very big error bar on that estimate, but I think we can start to potentially sense some interesting things. Now, all of this has been with large nanodiamonds, and so if you want to actually use these things as a sensor, say in an intracellular environment, you want to use small nanodiamonds so that your target are very close to your NV center surfaces, your nanodiamond surfaces. The issue with going from a 600 nanometer nanodiamond to a 30 nanometer nanodiamond is we drastically lose contrast. You lose T2 time, but the most significant loss is counts as well. So I think it's maybe 1% as bright per mass of the nanodiamond. So all of these things together mean that we are a lot less sensitive, and we're looking into methods to try and improve this. But yeah, we've kind of demonstrated these are the same DIR peaks. You can see in the kind of turquoise trace. We're still doing P1 center DIR measurements in 30 nanometer nanodiamonds, but it comes along with a few different issues. So yeah, we would our kind of goal for this is to sense external spins, and so we've done some simulations on how sensitive we would need to be to do that kind of detection, and we're not a million miles away. One of the current issues is that tempo degrades under optical illumination. So as we're trying to get more and more SNR for our tempo signal, we're also degrading it at the same time, which is a little bit tricky, but we're thinking about different management strategies for that. And yeah, so yeah, this is our summary. Yeah, essentially we're doing lots of nanodiamonds in maybe 500 by 500 micron area. You can use this for diagnostics as a spin label. We can make these systems portable and low cost. We want to understand where the diagnostic advantage in that is. And also we're looking at these kind of coherent measurements at zero field for biochemical systems and other material systems. So yeah, I think that's us. We have maybe 10 minutes to answer questions. So yeah, I think we'd be more than happy to do so.
Felix and Ben, thank you for a super interesting presentation. Definitely, it's a very big progress over the last year. So congratulations on scientific achievements. So we are open for questions. Please unmute yourself and start asking your questions. Maybe while people are getting oriented, I wanted maybe to bring a little bit more your discussion of role of nanodiamond size on your next to last slide, you have an array of nanodiamond sizes. Can it be, say, not 30, but somewhat larger particles, not to go to very bad, low extreme, but to optimize, like you did a lot of beautiful modeling, say, in other parts of your presentation.
This is actually a theoretical. This is also modeling work. This is a theoretical Monte Carlo simulation of what kind of relative contrast we can expect. And yeah, this is absolutely our plan for the next. After we get back from the Christmas break, we want to quantify our sensitivities with a range of different particle sizes, and then in combination with that and the distance of our molecules from the NV, we can work out where the optimum particle size is. Yeah, that's absolutely our next step.
And do you plan to play with modification of nanodiamond surface, like, say, these recently silica shells have been in favor in the nanodiamond NV nanodiamond community, demonstrating improved uniformity between particles, single particles, and also improved T1 and T2. Say what you think about thickness of silica shell if you would, say, use that approach.
I think it's the same. The same question again is I mean, we're looking at sort of surface modifications, chemical modifications as well, and we're interested in all of that stuff. Anything to improve the properties. And with the silica shell, I guess it's a trade-off just between proximity of the target to the NV center and the improvement in the potential improvement in spin properties. I don't know if you would add.
Okay, please, questions from the audience.
And we're also looking at, so in the hub, actually I forgot to mention this, in the hub we also have Ollie Williams and Gavin Morley, who published a paper on also on these sort of longer T2 nanodiamonds. So we're also interested in all of this kind of stuff. And I know you guys are working on that as well.
Are there any other questions, or I can continue to ask mine? As regarding your LFA part of presentation, so you did benchmarking with gold nanoparticles, right? At the same time, it is known that europium nanoparticles are commercial, say, competing technology. So it's probably better to compare oranges to oranges. Did you look at, say, fluorescent technologies to compare our approach, nanodiamond approach, since both of them would require additional investment into detection tools, right? Versus these gold nanoparticles with practically no additional device-related investments.
Yeah, definitely. I mean, so the figure from that paper that I showed near the beginning, they go into a lot of this stuff with and especially European particles, and there's some really good studies on European particles. We haven't done any direct comparisons yet, but yeah, it's definitely something we've talked about, and for sure it would be a really good comparison. Because they get really good sensitivities by doing this time gating and having these long persistence particles. Can you buy them commercially?
Yeah, you can buy them commercially. You can buy readers. So the point would be when you will come to the point of commercialization and start to approach your potential customers, where questions will be. It will be benchmarking with europium.
Yeah, thanks, Olga. Yeah, I mean, definitely this is something we should do.
Yeah, it's quite a market sector occupied by that technology. It's the closest competing technology.
I guess what in terms of any potential advantages, I guess sort of the main potential advantage that we have over europium nanoparticles. Well, I don't know how the sensitivity compares, but aside from that, because you can use nanodiamonds kind of as multimodal sensors, I think there are kind of interesting things you can do in terms of separating out specific and non-specific binding that you maybe couldn't do with European particles. But yeah, we're exploring that right now, so we haven't published anything on that yet, but hopefully in the next sort of year or so.
And also, it's excellent that you have your modeling tools. You can always, to do some runs with other particles with their density of surface binding sites and so on. So it's interesting if it's possible to
exactly. That's one of the main
insights.
For sure, that's one of the main aims of this work is to be able to do to get a new particle system, and we're hoping this, and we're framing it in a way which is generally applicable. And so what we're hoping to do is you get a new particle system and you do a very limited number of experiments, maybe an afternoon of experiments, and then you can extrapolate to a whole range of different scenarios. That's the idea of that work.
We have question. Sultan, please unmute yourself and ask your question.
Okay, thank you for your presentation. I just want to know if we could know the type of your nanodiamonds.
So could you repeat that?
The size or the type?
I just want to know the types of the nanodiamonds.
Types of nanodiamond.
Yeah, we're using Adamas nanodiamonds.
Okay, from Adamas. Okay. Also, could you please tell us about how you control these nanodiamonds, because they are very susceptible to noises? Is there
Sorry, what was the last bit, the last sentence? How we control them with what?
These nanodiamonds. How you control, because they are very susceptible to noises, and they have a lot of challenges, because you are also working here on nanodiamonds, and they are easily susceptible to noises.
Are you talking about noise in terms of sort of magnetic noise or things in the sample?
Or magnetic noise?
I guess one of the key ways to control for that is full sequence design that people have done fairly regularly in NV sequences. Like you have a reference pulse, or if you're doing a T1, control with a pi pulse, because there's lots of different things that can vary, your charge state over time and all that kind of stuff, or your laser fluctuations and things like that. So I think a decent amount of it is full sequence design.
And in terms of functionalization, when we're in more complex samples, we use diamonds from Adamas with a polyglycerol layer, and that gives us a number of advantages in terms of where mostly hydrophobicity, so a reduction in non-specific binding, and also some protection for the surface, and also I think it's probably good to have this very negatively charged surface, good for the NV charge state stability.
Okay, thank you.
And also you use quite large particles, right? So their subsurface NV centers are much more stable for these sub-100 nanometer particles. So probably there is some charge instability.
Yeah, certainly our NVs in the bulk are a delight.
Okay, do we have more questions? Oh, it seems everybody is happy and satisfied. So I would like to thank our speakers. Very interesting information. We don't care about that there were interruptions. And everything was resolved. So it was very, very impactful and very enjoyable lecture.
Right. So thank you so much for having us.
So much, and Merry Christmas, Happy New Year, healthy and prosperous in terms of scientific ideas, scientific innovations. I wish you all the best with your research in 2026.
Thanks so much,
And
Olga.
hope to meet at some conferences and host you in North Carolina.
Yeah, I hope so.
Okay.
Thanks so much. And thanks everyone for coming.
Yeah, thank you, everyone.
Okay, thank you everybody
Olga.
for visiting.
Thank you. Happy holidays.
You too.
Happy holidays.
Bye. Holidays.